Okay, so we all know a mother's health during pregnancy is super important, right? But what if I told you that a simple cold, just a temporary infection, could leave a permanent fingerprint on her child's brain? It sounds like science fiction, but it's not. Today, we're going full-on detective mode, following the molecular breadcrumbs to solve a genuine prenatal mystery.
And this is the big question, the one that's kept scientists up at night for years. I mean, how can an infection that's gone in a week or two actually change the fundamental blueprint of a brain for a whole lifetime? We've seen hints in the data for decades, but the how? Well, that's always been the missing piece. I mean, check this out. For years, massive studies have pointed to this really unsettling connection. Things like the flu or even intense stress from, say, a famine during pregnancy, they're linked to a higher risk of neurodevelopmental disorders like schizophrenia and autism in the kids later on. The link is there, it's solid, but the biological dominoes that have to fall to get from point A to point B, total black box, well, until now.
All right, so to crack this case, you can't just watch from the sidelines. You've got to get in there and experiment. But how on earth do you do that? I mean, this is incredibly sensitive stuff. You can't just give pregnant mothers an infection for science. Well, the answer, as it so often is in the lab. Scientists came up with a really clever way to mimic these conditions safely. It's called the Maternal Immune Activation Model, or MIA for short, and it's brilliant. Instead of a real live virus, they give pregnant mice a harmless substance that basically tricks their immune system into thinking there's an infection. It's like a fire drill for the body. This is key because it lets them study just the immune response itself without any other confusing factors and see how that alone affects the baby's brains as they develop.
Of course, before they could even start, they had to prove their model was legit. Did this fake infection actually work? You bet it did. Look at this graph. You can see this huge spike in something called interleukin-6. That's a major signal for inflammation. And you can also see the mice lost a little weight, just like you would if you were sick. This was the smoking gun. It proved the mimicked infection caused a real, measurable immune response. Game on.
Okay, so the model is solid. The stage is set. Now the real detective work could finally begin. Scientists could, with total confidence, look inside the brains of the offspring once they were adults, hunting for those tiny molecular fingerprints left behind by that immune event, way back before they were even born.
So, what did they find? Well, the clues they dug up were specific, they were significant, and they pointed straight to changes in the brain's most basic wiring.
Get this. In the brains of those adult mice, two things really stood out. First, they found way more of a specific serotonin receptor called 5-HT2A. And second, the actual physical shape of their dendritic spines was different. Dendritic spines are like the little satellite dishes on your brain cells that catch incoming signals. So basically, you've got changes to both the receivers and the wiring. That's a huge deal.
And this chart right here, it just lays it all out. Look at the bar on the right. That's the mice from our MIA model. The levels of those 5-H2A receptors are just through the roof compared to the normal control group. We're not talking about a little blip here. This is a massive, screaming molecular signal.
So what we've got are two totally different brain profiles. You've got your standard, healthy brain with everything in balance, and then you've got the MIA brain, which is just flooded with these extra receptors and has physically different connection points. The big question is, why? What's the mechanism? What's actually causing this to happen?
And this is where it all clicks into place. After putting all these clues together, the researchers figured it out. They uncovered the mechanism, and it's an idea that's so elegant and simple, they call it the broken brake.
Okay, so the main character in this story is something called the glucocorticoid receptor, or GR for short. Think of the GR as the brake pedal for the gene that makes those 5-HT2A serotonin receptors. In a normal, healthy brain, the GR presses down on that gene and says, "Whoa there, slow down, we have enough." So here's how the whole domino effect works. That infection during pregnancy, it actually damages the GR brake pedal. So now the brake line is cut. And what happens when you can't hit the brakes? The engine, in this case the gene making the 5-HT2A receptors, it just redlines. It's stuck on full throttle. This flood of extra receptors messes with the brain's wiring and really screws up its ability to filter out all the noise from the outside world.
But here's where it gets really, really exciting. Because if the problem is just a broken brake, well, can you fix it, right? Once you understand exactly what's broken, you can start thinking about how to repair it.
And that was the next big test. Could the scientist actually go in at a molecular level and fix this broken pathway in the adult mice? Was it too late? Or could they reverse the damage?
The results were honestly just incredible. They used a special molecular tool to basically give that GR brake pedal a boost. And look at this. It worked. They brought those sky-high 5H2A receptor levels right back down to normal. They literally fixed the brake.
And this wasn't just some number on a chart. This molecular fix had real, tangible effects. It restored the mice's sensory motor gating. Now, that's a fancy term for the brain's ability to filter out background noise, to tell what's important and what's not. And that exact function is something that's often a problem in conditions like schizophrenia. So they didn't just fix a molecule, they fixed a behavior.
Okay, this is amazing stuff in mice. But here it is, the billion-dollar question. Does any of this actually matter for humans? Does this whole broken brake idea mean anything for real-world human brain disorders?
Unbelievably, the answer seems to be yes. When the researchers looked at brain tissue from people who had schizophrenia, they found the exact same thing. The same broken brake. The same problem with that glucocorticoid receptor. I mean, wow, that's a direct, stunning link from the mouse model straight to the human brain.
This quote from the study really nails it. What these scientists have found is basically a master control switch in the brain. And it looks like when that switch breaks early in development, it could be a massive piece of the puzzle for understanding these super complex disorders.
So that leaves us in a pretty incredible place. We've gone from an old observation that something was happening in pregnancy all the way down to a single, specific, and, most importantly, repairable molecular pathway. And it leaves us with this huge, hopeful question. Could fixing this one broken brake be a future key to treating, or maybe even preventing, some of the most difficult and mysterious disorders of the human brain?